Hello, thank you for being here and watching this presentation. I will be talking about protein stabilization and delivery, a case study with invasion plasmid antigen D, or IPD as we like to call it. My name is Nicole Montoya and Dr. Mark Shiflett is my advisor at the University of Kansas. I am getting a master's degree in chemical engineering, so let's get started. Hello. The problem that we are trying to solve is related to the vaccine cold chain. The vaccine cold chain is basically the whole process of transporting and storing vaccines at 2 to 8 degrees Celsius from manufacturing to delivery site. Um, and because of this process, approximately 50% of vaccines are actually wasted. And some of the main problems in the vaccine cold chain are inadequate cold chain capacity, lack of functioning cold chain equipment, poor temperature monitoring and maintenance systems. And this topic is becoming even more important as we are living through the COVID pandemic. A lot of vaccines that are already in clinical trials require a negative 80 degrees Celsius freezing temperature at all time in order to retain stability and activity. So we are really excited that this work will be able to be translated and it has a very large need and impact. The problem that we The ultimate goal of our project is to develop and manufacture a syringe-like device that transports and stores silica-mobilized vaccines. And as we can see in this image here, the syringe will consist of 3 different chambers. The first chamber will contain a desorbing agent that's separated from the silica-mobilized vaccine by a membrane, and then the third chamber will be empty space. Once the plunger of the vaccine is pushed down, the desorbing agent will be mixed with the silica immobilized vaccine, and the free vaccine will go through the second membrane, and then that free vaccine will be ready for administration. The ultimate So here's a little bit of background in our project. We are using invasion plasmid antigen D, or IPD, as the model protein because it is an antigen protein in shigellosis. Shigellosis is a gastrointestinal disease, and over 1 million people die of this disease annually. This disease is more predominant in developing countries. In the figure on the left, we can see the proposed interactions between the IPD protein and a mesoporous silica gel. And as we can see, the nitrogens and hydrogens in the protein create hydrogen bonds with the oxygens and hydrogens in the mesoporous silica, and that is how we are going to stabilize the protein so it stays in place and does not unfold even when it is exposed to higher temperatures. In the right, we are just showing a couple of characteristics that make silica so attractive for this project, including high thermal stability, useful porosity ranges for proteins, simple chemistry, and a mild pH. And encapsulated biomolecular samples have shown to retain high activity and also a tunable surface chemistry. The first step in our project was to characterize different silica gel samples, and this characterization was done using BET, and the average pore diameter, accessible pore volume, and accessible surface area were found using BET, and they are listed in this table. Our experiments consist of 5 main steps: adsorption, washing, heating, desorption, and circular dichorism. So for the absorption step, 30 milligrams of silica gel and 0.7 milliliters of IPD at a concentration of 1.5 mg per ml are mixed for 20 hours. Then the supernatant protein concentration is measured. Finally, the silica is washed to remove the unbound proteins. Then the silica IPD complex is heated at 95 degrees Celsius for 2 hours. Then after that heating process, the proteins are removed from the silica with 0.7 mils of 10% LDAO, which is the detergent that we use. And finally, a CD analysis on IPD is used to evaluate the secondary structure. The adsorption process is governed by electrostatic interactions and hydrogen bonding between the hydroxyls on the silica surface and the functional groups of the amino acid side chains on IPID. The most abundant amino acid in IPID is serine, which makes up 13% of the structure. This creates the potential for a high level of hydrogen bonding between serine residues and the silica surface. There is a strong correlation between the average pore diameter of the size of the silica to the percent of IPID adsorbed. For example, silica sample S3 has a pore diameter of 15 nanometers and absorbed 91% IPID, while silica sample S1 has a pore diameter of 3.9 nanometers and absorbs an average of 9% IPID. Silicas with a pore diameter greater than 15 nanometers are expected to absorb more than 90% IPID. To determine the maximum IPID adsorption on mesoporous silica, the IPID adsorption isotherm was fit using the Langmuir adsorption model. The Langmuir adsorption isotherm assumes that all surface sites are the same, that all adsorption occurs on vacant sites, and the adsorbent interactions are negligible, and that all sites are either vacant or occupied. Furthermore, the Langmuir adsorption model also assumes monolayer chemisorption. In the figure that we have here, this is the Langmuir isotherm for our silica gel S3, and in the right we have the equations for the Langmuir model, the linearized form where Cm is the maximum monolayer coverage and K is the ratio between K adsorbed over K desorbed. The linearized form of the Langmuir model was plotted in the figure below, and the maximum monolayer coverage Cm was determined to be 181 milligrams per gram, and the equilibrium rate constant was determined to be 21 milliliters per milligram. From the close fit of the experimental data to the Langmuir model, it is possible to deduce that the adsorption of IPID onto mesoporous silica gels is occurring as a monolayer adsorption mechanism. Using a surface area for IPID of 112.8 nanometers squared per protein, and this number is based on using the largest two unit cell parameters of IPID, which are 10.07 and 11.28 nanometers. Using the molecular weight of 39 kilodaltons, it is possible to convert this maximum monolayer coverage from milligrams of IPID over grams of silica to meters squared of IPID over grams of silica. The reason why we want to convert the monolayer coverage to meters squared per gram is because that way we can compare the values obtained to the nitrogen surface area that was calculated experimentally using BET. So we can see that for silica gels S3, S4, and S5, the BET measurements are extremely close to the IPID surface area measurements. Comparing the Langmuir isotherm of silica sample S3 to the corresponding isotherm for silica sample S1, which the pore diameter in the silica is 3.9 nanometers, it is evident that the adsorbed IPID concentration for S1 is essentially negligible in comparison to the adsorbed IPID concentration for S3. The behavior displayed by S1 is expected because its pore diameter is smaller than IPID's external dimensions, which limits IPID adsorption. The plots in the left are Langmuir plots for silica samples S6 and S7. The IPID surface area for these two silicas is greater than the nitrogen surface area, suggesting that monolayer adsorption may be occurring rather than monolayer adsorption. IPID volume calculations are in good agreement with nitrogen BET volume measurements, supporting the hypothesis that adsorption is multilayer, for example, bilayer, in the silicas with pore sizes greater than 25 nanometers. Overall, if the average pore size is less than 15 nanometers, IPID cannot physically fit inside the pore and the adsorption is negligible. This occurs for silica S1 and S2. If the average pore sizes are between 15 to 25 nanometers, then monolayer coverage is achieved. If the average pore size is larger than 25 nanometers, then the absorption is multilayer coverage. The last and most important step in our experiments is to verify that the secondary structure is retained even after heating. So this plot shows three different curves. The first curve, which is the red one, shows denatured ipity. The green curve shows native ipity, and the blue curve is ipity that was heated while it was adsorbed in silica. The key results are that the adsorbed ipity after heat treatment and also desorption displays a very similar W-shaped CD signal as the native unheated ipity. And in this case, ipity was heated to 95 degrees Celsius for 115 minutes while attached to silica S4. All the work that I have talked about up to this point was recently published in a Langmuir article. So if you are interested in learning some more details, some of the methods, and seeing more experiments that we have also done, please feel free to check out this article and be able to learn a little bit more. Now moving forward, we want to work with influenza. We are interested in the influenza virus because every year between 390,000 and 650,000 people die of influenza. The influenza viruses are enveloped by a lipid envelope surrounded core, and the two main glycoproteins are hemagglutinin HA and neuraminidase NA. There are three types of influenza viruses, A, B, and C. A and B are the ones that are responsible for the human flu outbreaks. There are three different types of influenza vaccines currently in the market: the live attenuated, trivalent inactivated, and quadrivalent inactivated vaccines. And moving forward, we will be working with QIV because in the latest flu season, over 85% of vaccines administered were QIV. And QIV has a higher immune response than its counterparts like TIV. And QIV is also very sensitive to freezing and heating, so our technology will be extremely useful. We are really excited to get started on this project as well. This is a really collaborative project, so I would like to acknowledge and thank the people that have been working with us for a few years now. Dr. Bill Picking, Dr. Philip Gao, the Shiflett and Algar research groups, Dr. Ana Rita Moraes, Simon Velasquez Morales, Murilo Toledo, Kaylee Barr, Rihanna Roth, and Eric Hartman. And if you are interested in learning more about what the Shiflett Research Group does, please visit our website at www.shiflettresearch.com. Thank you so much for your attention. I realize that this year AICHE is not the way that we all would have wanted it to be, but I still appreciate that you took the time to listen and watch through this presentation. If you have any questions, please let me know, and I hope you have a wonderful day.